Okay, so now we're going to talk about what's called the dual visual system. It's the system that uses rods and cones for different reasons. And then we'll also talk about color vision. Okay, so let's begin with what's called your dual visual system, or your duplicity theory of vision. So here we have what's called the Photopic system. This system uses rods and it's good for sensitivity, meaning it can really detect low light, but it's not good for acuity, meaning you can't really get details using this system. And this system relies on what we call convergence of many cells down to a few cells. So here's an example of it. So here, let's assume that these are all rods, photoreceptor rods, okay? These are your rods. In this system, many rods will converge onto a common bipolar cell. Same here. These converge onto this cell and here. So maybe 100 rods will snap with the same bipolar cell here. Okay, and then these bipolar cells will synapse with a common ganglion cell. So these are bipolar cells here. This is your ganglion cell, which forms the optic nerve. It travels in the optic nerve. So make a point here. This is your optic nerve. So when light is very dim, many rods can pick it up because it's very dim light, and then these many rods can summate to create action potentials in the pathway. Okay, so many rods can pick up a very faint light intensity to still create a message that will eventually generate action potentials in the optic neuron. But anything that happens here, it will be unclear to you where it's coming from, because this signal may come from here or here or here or here. Anywhere in this field is game for turning on the optic nerve. So you can detect things, but you can't tell where it's coming from. That's called low visual acuity or visual details. So that's why this system, this Photopic system, is ideal for sensitivity, meaning low light conditions, but not good for really giving you specifics as to where in the visual field is coming from. So that is this Photopic system. Let's next do the Photopic system. This system is geared towards details, not so much towards sensitivity. Okay, so here we go. In the Photopic system, it uses cones only, and there is no convergence in this system. And here is ideal for acuity, not so much for sensitivity. Okay, so here, let's demonstrate it. So here, if you have cone, these are all cones. Each cone will be linked to its own individual bipolar cell. I'm almost there, okay? Which then will be linked to each bipolar cell in turn will be linked to its own ganglion cell, which forms, again, your optic nerve. So since it's 1 to 1 to 1, there's no convergence here, which means that if light is very low, it may not be able to activate any of these pathways because they can't summate together. because they don't share neurons along the pathway. So it's bad for low light conditions. This is why you use cones when it's bright outside or in the room when the lights are on, because that's when the light is strong enough to turn them on. So cones are good for bright light conditions, but the good thing here is that if this neuron is on, you know that the image must be right here. If this is on, it must be right here. So you can tell exactly where it's coming from. You have good details there due to the lack of convergence, but you have low sensitivity due to the lack of convergence as well. So that is your photopic system. Okay. We'll next do color vision. Okay. And color vision uses cones as well. Okay, so cones come in three types. So you have your blue cones, your green cones, and your red cones, also called short cones, medium cones, long cones. And these cones are different based on having a different opsin in their photopigment, okay? Keep that in mind. So the retinol is the same, but the opsin part is different. So it's a different protein, means a different gene that codes for it. In fact, while we're here, your blue cone, the gene for the blue cone opsin is found on chromosome number 7, and the gene for the green and red cones are found on the X chromosomes. Okay, so that's where you find the genes that code for these cones. All right, now, in order to detect color, we have to have a differential response of each of these cones to light. Meaning, your blue cones, for example, are optimally activated at a wavelength, let me show this text here, a wavelength of 420. So blue cones at 420 nanometers, they are maximally responsive. Then your green cones are at 531 nanometers, they are maximally responsive. And your red cones are at 558. So different light wavelengths will turn them on differentially. So basically, a different color light will activate them at different levels. And so the color you get, our color is based on what's called trichromatic color system we have, where the different levels of responsiveness of each cone gives you a certain color perception. For example, the color of, say, violet. So again, you have blue cones, green cones, red cones. To perceive violet color, here, half of your blue cones will be at 50% activation, green cones at 0, red at 0. This will give you the violet color. Similarly, if you do one, say, for red color is 0.005%. That's red color. Okay. Another one, the third one here, for green color is 85, 97, I believe, and 0. That is your green color. So all of our colors that we perceive is based on how these three different cones respond. Okay. All right, so while we're on this topic, then the idea is something called colorblindness. So colorblindness occurs when different cones are missing. You know, for example, maybe the gene for blue cone got mutated along the way, and so you won't get a blue cone gene, or you won't get a blue cone. So there are three common forms of color blindness. So the lack of green cones. So when you have no green cones, that's called deuteranopia. Deuteranopia. That's the most common form of color blindness. When you have no red cones, that's called protanopia. And the lack of blue cones, which is the least common issue there, is called tritanopia. Those are your three forms of color blindness based on the lack of a particular cone. Okay, we'll stop here. Pause here.